Hello, my name is Alejandra Baena and I'm a sophomore, pre-med sophomore studying biochemistry in the College of Arts and Sciences at the University of Pennsylvania. This summer, I had the honor of working in Dr. Irwin's digital neuropathology lab thanks to a program at our school called PERM. Our lab integrates immunohistochemical techniques with quantitative digital analysis tools to examine post-mortem tissue and improve the diagnosis of neurodegenerative diseases and clinical disorders. Frontotemporal lobar degeneration, otherwise known as FTLD, is the third most common pathologic diagnosis in neurodegenerative disorders, after Alzheimer's and Parkinson's. Of the proteinopathies that encompass FTLD, tauopathies, FTLD-tau and FTLD-TDP are the most common. My current project involves studying the pathological differences of these two main types of FTLD in a region of the brain called the Locus Coeruleus, since early evidence from our lab suggests that this brainstem structure plays a major role in arousal, learning, and other important cognitive functions, is more vulnerable to pathology and neurodegeneration in FTLD-tau compared to FTLD-TDP. Thus, finding an accurate way to differentiate these proteinopathies in living patients is necessary for clinical trials that plan to target these abnormal protein buildups. And the purpose of this research is to determine if the LC, Locus Coeruleus, is more vulnerable in FTLD-tau compared to FTLD-TDP. We examined post-mortem LC tissue from 48 patients diagnosed with FTLD-tau and 48 patients diagnosed with FTLD-TDP based on available tissue, and we generated ordinal score ratings for TDP and tau inclusion burden, as well as the extent of neurodegeneration in each group. Later, in a subsample of a FTLD-tau and FTLD-TDP subsample patients, we use digital imaging tools to quantify levels of pigmentation in LC neurons, resulting in measures of percent area occupied of melanin with LC neurons, in addition to the number of LC neurons with melanin overall, because we believe that the presence of melanin or the lack thereof could also be an indicator of neurodegeneration. And we discovered overall that higher extracellular pigmentation and depigmentation was identified in FTLD-tau compared to FTLD-TDP in the selected cohort. Similarly, LC neurons in FTLD-TDP had a greater percent area occupied by melanin and a greater number of melanin-positive cells than the FTLD-tau group. Our findings suggest that the LC is more vulnerable to abnormal tau inclusions compared to TDP inclusions, and that the greater burden of tau is associated with more neurodegeneration of the Locus Coeruleus in FTLD-tau compared to FTLD-TDP. So some future directions for my project include expanding our cohort in order to obtain more representative data and be able to draw more statistically significant conclusions, and using healthy LC tissue to develop better parameters for the digital imaging software. So we will be using this healthy LC as a control for our digital imaging. Additionally, we are also planning on using more markers, including glial and tyrosine hydroxylase stains, to better detect the type of cells where pathological inclusions are located. And overall, this experience has allowed me to learn a variety of techniques ranging from dry lab analysis of the digital images, basic coding and statistics, to wet lab immunostaining, in addition to strengthening my understanding of the field of neuropathology. In this lab, I have found a very collaborative and supportive environment that I know will help me grow and develop the skills I need to become a better researcher. As a young scientist, I truly appreciate having an environment where I can be myself and ask for help whenever I don't understand something. In this lab, everyone is always patient and helpful. My career goal is to become a neurologist, but I also want to explore the field of medical research, particularly the diagnosis of neurodegenerative diseases. Having the privilege of working in Dr. Irwin's lab was an invaluable opportunity to be involved with my career interests and directly interact with renowned experts in the field of neuropathology who have inspired me to continue studying this field. From clinical shadowing Dr. Irwin to observing brain autopsies and finding proteinopathies under the microscope, I have gained a better understanding of the neuropathological biomarkers of frontotemporal disorders. I will continue working on the LC project throughout the school year and perhaps pursue a more independent project in the future. Thank you so much for listening, and feel free to scan this QR code to access my poster if you would like. Also, feel free to comment if you have any specific questions about this project or general questions about getting involved with research or anything else you would like. Thank you.